Welcome to this webinar about how to access UniProt resources programmatically. It's part of the series EMBL-EBI, Programmatically, Take a Rest from Manual Searching. My name is Clemens Pichler, and while this webinar is using and talking about RESTful services, it does not provide a general introduction to those. For this, I would refer you to the first webinar in the series, which is linked here on this slide. The webinar also assumes some familiarity with UniProt in general and the knowledge base in particular, but it's not critical. If you'd like to brush up on your knowledge about UniProt afterwards, you can use some of the resources linked from this slide later on. We'll just jump straight into the resources that UniProt provides and which ones of those can be accessed programmatically. We'll then move on to demonstrate how to use those REST services by going over a couple of examples before finally summarizing what we've learned today. The UniProt Consortium provides many different resources. Front and center of those is the UniProt Knowledgebase, represented here by the blue-green box on the left, which is what most people refer to when they talk about UniProt. But next to this resource and underpinning it and connected to it are many other resources. For example, the UniProt reference clusters for sequence similarity searches, the UniProt archives of sequences, a special view on proteomes, and many supporting datasets ranging from literature citations to controlled vocabularies for subcellular locations. In addition to that, we also provide a small set of tools: BLAST, sequence alignment, ID mapping, and peptide search. Practically all of those resources and some of those tools are exposed via RESTful URLs on www.uniprot.org, and we'll use the UniProt Knowledgebase since it has the most sophisticated querying facilities as one example, and the ID mapping service as another example in the following because it comes in very handy. UniProt has also started providing many additional datasets, many of those large scale, which are not necessarily exposed at all on the uniprot.org website, but instead are only exposed programmatically via www.ebi.ac.uk/proteins/API. Those datasets and services cover, for example, genomic coordinates. This would allow you, for example, to get the genomic coordinates for a given human protein, for example, or the other way around. If you provided the genomic coordinates on a human chromosome, it would give you the UniProt IDs that map to that particular location. The services also provide access to antigenic regions mapped onto human proteins imported from another resource, data about protein interactions, sequence features, natural variants, proteomics data, protein isoforms, and taxonomy as it is used in the UniProt Knowledgebase. We'll use one feature of the proteomics data services as an example here because it allows us to emulate the peptide search, which is a tool available on uniprot.org, but not exposed programmatically there. Let's move on to the first example, querying the UniProt Knowledgebase. The REST endpoint for this service is www.uniprot.org/uniprot/. It uses the HTTP method GET. The documentation for the parameters for this service are linked here in the middle, ending in API_queries. And the pro tip about learning these parameters is to use the interactive advanced search on uniprot.org and have the corresponding URLs constructed for you, because URLs is what RESTful access is about. So how would this look like in real life? Say if we went to uniprot.org now and opened the advanced search, we could launch a query that targets the field protein name with the term polymerase alpha, the taxonomy section with the term mouse, which is short for mouse, and we could filter for reviewed entries only. If we then clicked the little magnifying glass icon in the bottom right, the search would launch and it would be translated into a plain text version of the search, as well as display the corresponding URL in your browser's address bar. You can immediately see that the endpoint, the REST endpoint that I mentioned on the previous slides, www.uniprot.org/uniprot is in this URL as well. The rest looks a bit like gibberish, but we'll get to that in a minute. Obviously, the actual results of the search would be displayed on the website as well. This way of learning about fields and parameters is valid for all the RESTful services exposed on uniprot.org. How can we use this in a program? For this, we use Python, and we start by importing a package that's called requests, which is a package to help communicating with servers in general, and in this case, the UniProt servers. We copy the URL that we've used or generated previously by our search on uniprot.org and copy it into a variable called full URL. The name doesn't really matter. There's one tiny modification we make, which is we append format equals list to the very end. And then we pass this URL to the request package's get method. Remember that I initially said the UniProt endpoint has or uses the HTTP method get. And then we get the results. If the results are okay, we print the output. The output is provided as a string, meaning text. That's why we print result.text in the code. And you see the output at the very bottom of the screenshot. It's 4 different UniProt accessions, which is what we specified in the format.equals-list that we appended to the URL. This is encouraging, so it seems to be working. But using the full URL like this is not particularly nice. So maybe we can change this. If we reuse the code from the previous slide, we can extract the base URL, the REST endpoint for the UniProt Knowledgebase, uniprot.org/uniprot/, because that's the part that stays the same for all the queries that we launch searching the UniProt Knowledgebase. The rest we call the parameters. It still looks ugly. It's very hard to read and even harder to write by hand. But if we combine these two, the base URL and the parameters, we could run the code again and we would get the same output as you can see at the bottom of the screenshot. Still, we'd like to improve how we have to pass the parameters to the function so that we can do it in a more human-like way. Note here that the parameters have 2 parts in it that start with key equals value, and we'll use that to restructure the code. What we do here is, again, reusing as much of the code from previous slides as possible. We construct a Python dictionary with 2 keys in it, one called query, now with a value where we can type in the search phrase in plain text, and another key called format that has the value list, what we used before. We store this in a variable called payload. It could be any name, doesn't really matter. And then afterwards, we pass this dictionary to a parameter for the requests get function that's called params. And we run the code again, and we'll see the output is still the same. So it seems to have worked. If you'd like to check what has been going on behind the scenes, you can look at what the requests package has constructed in terms of the URL. At the bottom of this slide, you see if you type result.url, you will see the result that the package automatically constructed for you, and it's what we had before, what we copied from the uniprot.org website. Now, up until now, we've used format.list to retrieve a list of UniProt accessions. What if we wanted to use another data format? This now becomes very easy. We just change the value for the key format in our payload dictionary to, say, text. And if we don't do this, rerun the code, what we get is UniProt entries in plain text format. This obviously is only the very top bit. The screenshot is truncated at the bottom. So this is now very simple. There are many different formats for data retrieval. We've already encountered list. We've encountered text. There's also XML, which downloads UniProt entries in XML format, which is a format really that's meant for consumption by computers rather than humans. Then there is tab, which is short for tabular, and it allows downloading selected fields in tabular format. This requires an extra parameter called columns to specify which fields we'd actually like to download as part of the data. Furthermore, there is a format called FastA, which downloads canonical sequences of the query hits in FastA format. We can also download isoforms, which requires an extra parameter called include yes. Then there is GFF, which downloads annotated sequence features in a format called GFF. And there is RDF, which is similar to XML. It's meant to be consumed by computers. These are data formats available for the UniProt endpoint, so querying the knowledge base, but the formats vary depending on which REST endpoint is used. And so there might be others, or there might be fewer or more. I'd like to come back to the tabular format for one moment, because it is very powerful in that it allows you to specify exactly which bits of the data of a UniProt entity you'd actually like to download, and so allows tailored data sets. We can reuse the code which we've used before, again, modifying the payload dictionary that we had before. We keep the query as value for the key format. We now use tab, and then add the extra required key, which is columns, by providing a string of comma-separated field names. The field names you can find in the documentation that was linked earlier. Once we've done this, the rest of the code remains the same, and we print the resulting entries. And you see at the very bottom, we get tabular output with column headers boxed in green, and then 2 rows, the others are cut off, of data. They're wrapped because the lines were too long for the display. Yeah. So that's quite a powerful format to use. With this, I'd like to move on to the next example, which is using the identifier mapping and batch retrieval services. The REST endpoint for this is www.uniprot.org/upload-lists/, and again it uses the HTTP method GET. There are 2 documentation pages for this that provide all the details necessary. What does this allow us to do? It allows us to map UniProt accessions to accessions from other databases, and the other way around, map identifiers from other databases to UniProt, and there are very, very many databases covered in this service. It also allows us to retrieve UniProt entries in batch by just mapping from UniProt to UniProt. What it doesn't allow us to do is to map directly from one database that's not UniProt to another database that's not UniProt. We can circumvent this by first mapping from database A, which is our source database, to UniProt accessions, and then from UniProt accessions to database B, which is our target database. And that way we can still map from A to B indirectly. Let's look at how this works. Again, we use the requests package in a Python program, but now we're stepping up our Python programming a little bit, and we define a function to make it reusable. The function is called mapRetrieve, shown in blue here. The main name really doesn't matter. The function takes 4 parameters, one of them obligatory, which is the first one. It's called IDs to map, which contain those IDs we'd like to transform into those of another database. And then there is 3 optional ones, which have default values. There's source format, which always defaults to UniProt accessions or IDs, target format, which also defaults to UniProt accessions, and the output format, which defaults to list, a list of accessions. The base URL in this case is the REST endpoint for the ID mapping service, and we still use the now familiar dictionary payload, just with different key value combinations. We have the key from, which is source format, to target format, which is output format, and the query, which contains the IDs that we would like to map. Again, we pass this to the requests_get method and run the code that really doesn't look that much different from what we used before. We make one change though. Instead of printing out the results, we return them so that we can either print them out or store them and reuse them afterwards. Let's see this in action. Here we use a call to the mapRetrieve function we defined on the previous slide and pass it the mandatory argument of IDs that we would like to map. These are 4 PDB IDs. PDB is a database about 3D protein structures. And so we specify the source format as PDB ID. Again, how these source formats are to be specified is documented on the web pages I linked earlier. The output format, we choose text because initially we would like to see the corresponding UniProt entries in full. We store this in a variable called UniProt ACK, which when printed looks what you see at the bottom of the screen. Again, this is cut off and is only the top bit of the hits. But more interesting than just retrieving entries this way is what we talked about earlier, mapping from database A via UniProt to database B. So, as an example, we'd like to map from PDB IDs to ensemble IDs via UniProt. We start by only slightly modifying the code that we had on the previous slide. Again, we call map retrieve with list of PDB identifiers. Source format stays the same, but we leave away the parameter output format because it defaults to list anyway, and that's what the format we'd like to have here, a list of UniProt accessions. If we print out the resulting accessions, as you can see, there's 4 UniProt accessions which correspond to those PDB identifiers. Now, in a second run, we use those 4 UniProt accessions as input for yet another call to map retrieve, this time specifying target format as ensemble, because that's what we would like to map to. Source format we don't have to specify because it defaults to UniProt accessions, which is what we have as input here. The code looks a little bit funny because, as I noted earlier, what's returned by those services is text. And so it has new line characters in it, which we strip out, and then we split the string to generate a list as input. But this is just a little aside. Again, we store the results in a variable that we now call ensemble, and if we print ensemble, we get 4 ensemble gene identifiers. So, in the end, by chaining 2 calls to the map retrieve function that we defined earlier, we were able to map 4 PDB identifiers via UniProt identifiers to ensemble gene identifiers, which is good. Now I'd like to come back to those large-scale datasets exposed on ebi.ac.uk/proteins/API, or Proteins API for short. The nice thing about these services is that that page, the URL that's on top here, also contains the documentation for all these services. Going to that URL shows us a page like this, the Proteins API, that lists all the different endpoints or categories that provide different endpoints in this section. We'll focus on the proteomics section, and within that we choose one particular service that's called proteomics as an example. The actual REST endpoint for this would then be www.ebi.ac.uk/proteins/API/proteomics, again using the HTTP method GET. Now, the documentation here is not just one to read, but is constructed in a way that allows you to interactively explore and test the services at the same time. So what you can do here is you can choose the response content type, which is basically the data format that you would like to have, which is set to application JSON here. Among the many parameters available for this particular REST service, we choose one which is called peptide, down here, to provide peptides which are then searched against all the UniProt sequences and retrieve those that have a match for it. You can now test this by clicking on the little green button at the bottom that says try it out directly in the documentation. When you do this, what you get is, for once, the response body. This is the actual result of the query. It looks a bit like a Python dictionary. This is the JSON format that we specified as the result type. It contains at least one entry that's named DUS1 human, including its entire sequence. And this entry has at least one hit for one of those peptides we provided, which starts at position 281 and ends in 289. There are other hits, as you can see by the slider here, which were cut off again in the screenshot. But in addition to just the results, the documentation also auto-generates the URL we could have used in a programmatic access for this. And in addition to that, it actually generates sample code in different programming languages as well. Here is Python, and as you can see, it also uses the requests package, which is nice since we already know about it. And to see how well this code performs, we can now copy it and paste it into a Python interpreter. Doing this, we'll see The sample code uses, again, the full URL like we did in our very first example, so it's not very nice because it's not readable. And then there's one little difference, where in the previous examples we used format equals something to specify the format we would like to have. It's done differently here. It does it using the HTTP header in a dictionary, in a key value, a pair called accept and then application JSON. This doesn't really pose much of a problem for us because the requests package allows us to pass just another dictionary, which is the dictionary here, to another parameter called headers, and then we can just run the code, and we get the same results as we had on the previous slides, just not formatted as nicely, but that's due to the interpreter I've used for this screenshot. But now we have standards now. We've already improved the code that we wrote previously, so we can improve on this code as well. Again, what we can do, we can extract the base URL, which basically is the REST endpoint for the service we've just used, and that's up here, the base URL at the very top of the code that ends in API/proteomics. And then we can use our familiar payload dictionary again, this time with the key value pair peptide, and then we provide as a value the comma-separated peptides that we want to search in this service. And we just pass it again to the params parameter in the requests get method. And we keep the headers parameter as well. When we run this, at the bottom you can see we get the same output. So this is just much more usable and user-friendly and does the same job. With this said, I'd like to summarize. So UniProt provides many different RESTful service endpoints using two base URLs. One is www.uniprot.org and the other one is www.ebi.sa.uk/proteins/API. You can learn about and explore the former via the advanced search on uniprot.org and the latter via the interactive documentation on that same page. If you need further documents and help, you can find them on the links that I've listed here. And if all else fails, questions can be sent both about content as well as about code to help@uniprot.org. With this, I'm at the end of the webinar. I'd like to point out that the series is not yet finished. There's another one coming up. Incidentally, it is about PDB-E, which is the database whose identifiers we've used in the mapping exercises. If you think of any questions later on, send them to help@uniprot.org and we'll be quite happy to answer them by email. Thank you.